Let's talk about the different types of mutations we're going to see in biology. Remember that mutations are permanent changes in the DNA sequence of a cell. Our first type of mutation we're going to talk about is a point mutation. These are changes of a single nucleotide within the DNA. Now, there are many different types of point mutations, but they all revolve around the fact they change only one single nucleotide. This can be like changing an adenine to a cytosine or guanine to a thymine. Our first type of point mutation is a substitution mutation. And just like the name implies, this is when one nucleotide is changed to a different nucleotide. Now, there are many different types and causes of these substitution mutations. But again, they're just when one nucleotide changes to another. A type of these substitution mutations would be like a missense mutation. This is when the mutation causes the amino acid to change. You can see in our illustration here, the original sequence of DNA produces the amino acid lysine. However, you can see in each of these types of missense mutations, a different amino acid will be produced by that original codon. The two different types of missense mutations are called conservative and non-conservative. This doesn't have to do anything with politics. A conservative missense mutation is when the properties of the amino acids will remain the same. You can see here in our conservative mutation that arginine has the same properties as lysine. In a non-conservative mutation, the properties of the amino acid will change. Threonine here has a different property than lysine, which means that's probably going to make the protein fold a little bit differently than if lysine was there. However, both of these are types of missense mutations. Our next type of missense mutation is a silent mutation. This occurs when the mutation doesn't actually change the amino acid that it codes for. So since the amino acid sequence doesn't change, the protein's not going to be affected. You can see in our mutation here, TAT and TAC both produce the amino acid tyrosine. So in this case, the protein won't be affected because the amino acid sequence is the same. In a nonsense mutation, the mutation is going to cause an amino acid to change to a stop codon. When this happens, the ribosome is going to stop translating the protein, which means the protein's shape is going to be dramatically altered. You can see in our illustration the lysine being changed to a stop codon, which won't produce any amino acids after it. Our last main big type of mutation is called a frameshift mutation. This occurs when one or more nucleotides are either going to be added or removed in the codon sequence. And as you can see, this shifts the frame of the codons in DNA. And that changes all the amino acids after that frameshift mutation, which means there will be a more significant change to the protein since all of the amino acids are going to be different. So these frameshift mutations can have a much greater effect on proteins than just a point mutation can. I want to mention here that these insertion and deletion mutations fall under frameshift mutations, and they are literally what they sound like. Insertion mutations are when you add one or more nucleotides. Deletion mutations are when you remove one or more nucleotides. And again, this changes the codon sequence that the ribosome is going to read, which then changes the amino acids of everything after the frameshift mutation.